today it will be divided. So we'll start with some water quality and then go to the lab, try to analyze it, then come back, talk about solids, then go back to the lab and try to get all the results, or read again these experiments are over time, read again, and I'll talk all about it as we go, or later in this talk. It is going to be a busy day, so crush whenever you crush, and we'll just if it can be interactive, then it'd be I guess easier to absorb. And the idea is a bit, I don't know if, I think it's a bit different than what we are used to, but I'll try, or let's start, I'll take a step backward and say that my background is fish, and aquaculture, water treatment in aquaculture, in pond aquaculture. That's where I studied. Then I studied soil and water sciences, biology, water and soil sciences, and then moved into civil engineering and water treatment. So, and in recent years, came back to aquaculture as part of what we are doing in the lab, which is waste to resource. So if to put a title on my lab, that would be the thing. And aquaponics gets into that overall scheme of waste to resource. In this case, fish waste into a resource and water reuse and things like that. So my looking at these systems is really like a wastewater treatment plant. I don't care about the fish, sort of speak, and I don't care about the plants. I care about water reuse, efficient water reuse, and water treatment. And looking at that from that angle brings, I think, a different perspective to the thing. So if we have a fish tank, that's a producing waste. Okay, they producing nutrients, but they produce the waste. And this is a wetland that treat the water, so it's a wastewater treatment plant, and each bit is like that. Okay, and therefore, in order to have high control or maximal control on each unit, we're looking at this system as a bunch of treatment loops or treatment systems. So this is one unit, and the plants is another loop, and the solid treatment is another, another scheme. And that's the way and that's the way we're treating that. But before we start and talk about treatment principles, so to put a baseline on the water quality that, and all obviously has to be within the constraints that it has to fit the fish and it has to fit the plants, right? Because we cannot do anything we want, but so the water quality has to meet certain standards. Now, it can be environmental standards when we're talking wastewater treatment, or it can be standards that appropriate for the fish and for the plants. So that's the deal. So we'll start with basically a list of the water quality parameters that are suitable to grow fish. So overall, we'll talk about water quality and general criteria, and water quality design target. We'll talk about the nitrogen cycle as probably the most important cycle. We'll talk about solids a little bit, and removal and treatment, because first you have to remove the solids away from the system, and then you would like to treat them. And we'll talk about just an example of aquaponics. So simply enough, there is one source of pollutants or nutrients or whatever, one source of input into this system, and it's food, fish feed. That's the only input in aquaculture systems that are recirculating or closed systems, right? There is no external inputs, where in a pond you'd have runoff and rain and God knows, dust. Here, it's in a closed container, there is one input, it's the feed. And the understanding the fate of this feed helps a lot to move forward and understand what the needs are. So when we have we introduce feed, and usually it's at a certain rate, sometimes 1 or 2 or up to 3% of the fish weight that we have there. Feed, unlike land animal, contains a lot of protein. So it's anywhere between 22 to 60% proteins. That's the range. And that's very unique because the carbon to nitrogen ratio in this feed is very different than waste, general waste, human waste, manure of animals and so on. That means that the treatment has to be adapted to this sort of situation. And it contains usually about 1% phosphorus, sometimes less, sometimes a bit more, but that's about right. And it contains about 30 to 40% carbon. That's basically what it is. And then there are some fillers. Yeah? So about 90-95% is eaten by the fish if you're efficient in your feeding. Now it's really important whether you feed 1 time a day, 3 times a day, or continuously over, with these automatic feeders. It will, the source will be different into the water, and that will also dictate at times the efficiency of your treatment. About 20 to 30% of most elements are absorbed by the fish, which means that 70 to 80% is wasted. That's a big chunk of material that is wasted there, nutrients, carbon, and phosphorus, and others, potassium. Of the eaten feed, about 20% goes to feces and require oxygen. Okay, so in the water we produce basically CO2, ammonia, and phosphorus. Of the absorbed feed, 70 to 80% is respired, okay, or excreted, again is ammonia and phosphorus and carbon dioxide, and only 20, 25, sometimes 30% is harvested by the fish flesh by itself. That belongs to a pond. It's not so much belong to this recirculating system. Now, as we said, the sole source of nutrients is NP and carbon, and the same thing with solids, obviously. So in order to move all these things and to keep the fish alive, we need oxygen. It's an expensive bit of the operation, and it's important bit of the operation, and it's very famous. Anyone who grew fish knows that without oxygen they die, unless you grow catfish or pangasius or God knows the different African catfish, that can breathe air. But basically it's that, and really best conditions is anywhere above 5 milligram per liter. And here, what I thought to do, and maybe one word of preface that I should have said, we're trying, I don't know how much we'll manage to do today, but we're trying to make everything quantitative and try to look at all these reactors in a quantitative manner, so it's all balanced and we know exactly what we put in in terms of energy, in terms of aeration, how much air, and so on. So everything has to be measured, basically looking at it as an engineering project, okay? Rather than a biological system in a way. And I think, again, that's a bit, it's the correct approach for these intensive systems. Okay, so the major thing is oxygenation or aeration, and each one of these devices that are used to aerate the water has different aeration properties and capabilities, and we need to measure or understand these capabilities of each one of these machines, and that's through an oxygen transfer type experiments, and we will not run these today. And I'll go quickly. I don't know if we will not go through all these calculations, but unless someone wants, and then I'll stop. So really, you know, when the differences, the aeration would work best when there is a difference between the oxygen saturation in the water, I mean, the actual concentration of oxygen in the water to the maximal saturation point. So if we have 1 milligram per liter in the water, the devices will work more efficiently than if we had 5. If we wanted to lift the oxygen from 1 to 2, it'd be easier, sort of speak, than if we wanted to increase the oxygen from 5 to 6, okay? Because of the difference in partial pressure. And this is something you can see in these figures. So it first be quicker, and then it turns and it levels, okay? So if we want to look at this, when we can determine these equations and figure what would be the line for each one of these devices, and it fits the you know, the rate of gas mass transfer, that's here, equals to some sort of a coefficient on diffusion of gas, and the area through which gas is diffusing, and the difference is between concentration and saturation and the actual concentration. So that's why it's a logarithmic note, and we can run these experiments where we reduce the oxygen concentration in water to almost 0, and operate our device, aeration device, and see how quickly, or measure the concentration over time, and create these graphs and get all these constants that can tell us what is this aeration oxygen transfer rate of our device. And again, these are used for ponds, but diffusers are used in our aquaculture. Liquid oxygen often used, aerolators. It depends on the size of the systems, whether it's urban or not. Okay. The other thing that is important, but often ignored in intensive aquaculture, is the carbon dioxide. And carbon dioxide, when you grow fish at low concentration, it's not an issue. But when you intensify and you grow fish at 100 kilos per meter cube, or up to 400 kilos per cubic meter, then it's becoming more important than oxygen. It's harder to remove CO2, to strip CO2 from these intensive systems than to supply oxygen to the fish. Okay? And why do we care about that? Because at certain concentration, CO2 has different toxic effects, just like on human. So it has narcotic effect. At higher concentration, it causes death to the fish. Calcium deposit in the kidney because it reduces the pH, right? It's an acid. So there is an acidosis that it causes, and reduces the affinity of O2 to the hemoglobin, and often, very often, it's ignored. And that's a limiting factor for intensification, okay? So it has to be stripped out. And again, only in intensive systems. I haven't seen the ones that we saw here, like 40 kilos per cube, is not an issue still. It's only when you go up with the situation. In terms of metals, that's another whole field that usually is not looked at so much, but should be considered, and specifically if we're using marginal waters or other waters than sea or fresh water from, clean fresh water. But if you want to use brackish water, groundwater, all sorts of different waters, if you want to work in Africa and you take water from a contaminated river, all these things becoming more of an issue. And we have some of these metals are also in the feed. And remember, fish are hyperaccumulator of many of these metals. So, it's, if we don't pay attention, we may end up eating it. So in general, it's toxic to fish at very low concentrations. Solubility increases with pH when pH decrease. Okay, so that happens with most metals, iron and leads and all these cations. The effective concentration changes obviously with species, but also with temperature and alkalinity. Okay, so aluminum, just rule of thumbs, and it's very different from one growth to the other, but it's usually considered to be, or should be, fairly low, and not so much in recirculating aquaculture system, but in fish ponds. Sometimes we use alum as flocculants to reduce algae or to reduce suspended material, and is in a wastewater treatment approach that can be used at times as a coagulant and flocculant, or calcium hydroxide. So these are the kind of things to look at the chemicals that we're using, or if we use copper for disinfection and things like that, then copper sulfide, then we have to pay attention that they're not increasing in concentration. This group of heavy metals, arsenic, barium, lead, iron, zinc, are alkalinity dependent. So as pH decreases and alkalinity goes down, their toxicity is much higher, because it's not often calcium and magnesium, which are divalent ions, can reduce the toxicity of these metals, okay, because they compete on the uptake sites on the gills. Calcium concentration ranges, but really we need calcium. It's a major element. We need calcium in the water, and for the fish we need magnesium. These ions are less alkalinity dependent. Sodium is a bit ridiculous in a way, because it depends on the fish. Fish can live in salt water or fish can live in fresh water. So this is freshwater fish, usually do not tolerate much higher than 75, but that's really, I put it, but it's not. And then we often talk, when we're talking aquaculture, we're talking about salinity. And as a measure of salinity, we talk about conductivity. And I'll talk about that a little bit as we go. But, but when we say salinity, the composition of the ions are really important, and I'll say that, a few words about that. So let me go here. So salinity is a measure of the sum of ions and cations in water. That's what it means. When we say salinity, 500 milligram per liter, we mean that if we summarize all these in milligram per liter, that would be the salinity. But it can be all sodium chloride, or it can be all magnesium sulfate, or it can be a range of ions. And that's crucial when you grow fish. So salinity is not a good measure unless you're talking about natural water or seawater or diluted seawater to talk about fish growth. You know, if we use brackish water, and that's something a lot of times happens in arid areas. We don't have water, natural water, so we go down to the ground. And then the ratio of ions is very different, and the fish are doing bad or good according to the ion concentrations in this water. And we often notice that potassium was one major, for example, ion that was missing in many groundwater. Okay. Okay, that's the importance of calcium, but we talked about that. So that's what we were looking at, sources of, or just talking about sources of salinity. And in nature, salinity is either from weathering rocks or from some chemical reactions and redox. And redox is reduction oxidation potential. You know, some acid-base reactions and so on, and precipitation. So that's in nature. In aquaculture, it's the original source of our water, and again, the food, or the fish feed. That's the sources of salinity into our system. And in the fish feed is not always balanced. And just to demonstrate the point that I was making, we ran this experiment with shrimp. And we use sea salt as control. Can you see? And that was first diet. These are the diets. I mean, not the diets, the water quality, the different treatments. This is sea salt and the breakdown of the major ion composition. Sodium, potassium, calcium, magnesium, total dissolved solids, which is basically the salinity. You know, and what we saw, the number of animals, what we saw that we had 96% survival. That was shrimp. Okay, when we composed our own salt solution, yeah, which was fairly similar to in terms of major ions to the original seawater, yeah, we already got much less survival, and we started playing with the ratio of potassium and sodium. We got all kinds of survival rates in these tricks. So there is something in the composition of seawater, and that's probably evolutionary thing that helps survival. Or it's not always just the major ions that determines you know, survival or capability of fish or shrimp in this case to survive. Okay, so the composition is very important. Another term that is often used in aquaculture in specific, but it's not very important in aquaculture, I guess, and because it's mistaken or confused with the term of alkalinity, is the hardness. And hardness is the sum of divalent ions in water, yeah, calcium, magnesium, manganese, and so on. But calcium and magnesium are the most prevalent ions, divalent ions in water. Okay, so and this index was developed by wastewater treatment people, okay, because of scaling issues on pipes and in boilers and things like that, where calcium precipitated when you hit the water. So for whatever reason, it's also used in aquaculture, but for no good reason. So normally, experimentally, it's anywhere around 100 or sometimes a little bit slightly above 100 milligram per liter would be important for the fish. Anything about 50 milligram per liter really in terms of calcium. And the ranking that people made for soft and hard water is the concentration of calcium and magnesium in the water. So anywhere up to 75 would be soft water, like rainwater. These would be moderately hard, this concentration harder and very hard. And here, as I said, it caused problems, but not so much for aquaculture. Okay? Why it is used, after all, if it's not relevant? Because often alkalinity, which is a very important measure, is contributed with calcium carbonate from limestone. And then there is a correlation between the amount of calcium and the amount of calcium carbonate. Okay? So it sometimes overlaps, not always. And it's not only calcium carbonate, it's sometimes dolomites, which is calcium magnesium carbonate. Okay? So let's move to what's more important. Not yet. It's the next slide. We talked about salinity, and the other way to describe salinity is total dissolved solids, which is basically ions, and that's basically similar to salinity. Okay, so we call it TDS, total dissolved solids. I put the TSS here, which is total suspended solids. In Australia, for example, it will be called NFR, non-filtrable residues, so it depends where you are, because sometimes in aquaculture this is also total soluble salts, and in plant, I think studies I read that it's total soluble sugars. So just make sure we know what we're talking about, but here it's total suspended solids, and that's the term known in wastewater treatment and aquaculture as well. And alkalinity anywhere between 50 to 300, and there are quite a few studies that shows that alkalinity below that affecting badly the fish performance, okay? And for certain reasons. First, because alkalinity is a source of carbon, because often it's coming, as we said, from calcium. Here. I haven't written on a board since I was a kid in class, in school. So, so it's, coming from calcium carbonate. So it's a source of CO2, of inorganic carbon, and that's really important for many bacteria, and specifically in aquaculture, who likes to use inorganic carbon. What process? Nitrification, yeah. So that's the deal. So it's a source of CO2 or carbon for for nitrifiers. It has a buffer capacity. I'll talk about that in a sec. And most often is a calcium and magnesium source, and it reduces metal toxicity. We talked about that a minute ago. pH is also very important here because we're talking about buffer, so I just put for the purpose of, so we have it, the definition, you know, it's for this purpose, it'd be well defined if we look at pH as the minus log concentration, molar concentration of hydrogen. Okay, so so really pH represents the amount of hydrogen ions we have in the water. Okay, that's on a logarithmic scale. That will be an important issue later a bit. Let's continue and stay with the natural alkalinity system in most natural water, which is calcium carbonate, and it's a pH-dependent thing, right? So at the lower pH, or it's an equilibrium, right? It's a chemical equilibrium system where we have H2CO3, okay, which is the acid form, but this is in equilibrium with CO2, okay? So this is a very unstable form of acid that in equilibrium with bicarbonate, HCO3, which is in equilibrium with carbonates. Okay, so it's a system. It's a buffer system, and it can absorb hydrogen ions or hydroxyl ions and move from one form to another and actually maintain a pH. So at lower pH, we have only CO2 or only H2CO3. Okay, and at the higher pHs, you see at around pH 6 point something, we have 50% CO2 and 50% bicarbonate, HCO3 already. But at pH of 8 or 8.3, everything is in the bicarbonate form, and we have none carbonates nor CO2. And above that, we start losing bicarbonate and get carbonate ion. And that's how this system is moving. So it's really, really an important and issue in aquaculture, and very important to identify it and make sure that we do not forget to add alkalinity, because there are some processes that consume alkalinity or produce alkalinity in the water. For example, nitrification consumes alkalinity, produces acidity. Ammonia, when it, when it's, we'll talk about that, but when it's converted to NH4, it's produce hydroxyl ions, so it's producing alkalinity. And denitrification is producing alkalinity, okay? So there are all these sort of important processes that produce or consume alkalinity, and it has to be maintained. pH, also, anyone who works with fish knows this figure, but for the purpose of making a baseline, I put it so low pHs, they're death points, and less reproduction, slow growth, best growth of fish, slower growth again, and then they die. And here is an example which is important for the importance of alkalinity, specifically in the, after looking at the pH table, then you can see that, that's in a pond. You're familiar with that? Anyone? Why pH would change in these ponds? It's just pond water. Yeah, algae, exactly, phytoplankton. So you're all familiar with that, or I don't know if all. But the concept is that, I'll say in a few words, algae or micro phytoplankton in pond respire, then they consume CO2, do photosynthesis, respire, then they produce CO2, do photosynthesis, so they consume CO2. CO2 is an acid, so they produce acid or consume acid in the, over the day. When there is light, they consume more than they produce, so pH would go up. And when they, at nighttime, when there is no photosynthesis, they'll produce more and consume less, so pH would go down. And in low alkalinity ponds, it can go down from really low pHs. This is not really low alkalinity, but it can go down to, the lowest it can go down with the carbonic system is about 4.5 or 5. That's where it goes, and it goes up to over 9, sometimes 10, and then can go back. When you have the alkalinity, it is moderating these bumps. And obviously, like we saw, the fish do not like these changes and do not like to live in changing environments. In terms of phosphorus, in this grocery list of water quality parameters, it's not important to the fish so much. It's important to the plants, and it's important to the environment more than anything. Phosphorus can cause eutrophication, is the most limiting factor in nature, and so on. But in this recirculating system, it's less of usually it's less of an issue until the water is being released into the environment, okay? So, so that's something to keep in mind, and it's of great interest in Europe, in the US, in different places because of the environmental considerations and the regulations that really making it's very hard to release aquaculture water into the environment. So that's where it's coming from. That's why it's of an interest to the industry. Before that, it was just went to the river. Nobody cared about phosphorus in the past. Nitrogen. So obviously nitrogen, because we get this 50% often feed, nitrogen is the most important, I guess, nutrient in this system because it's in high concentrations, and there are different forms that are toxic, like ammonia and nitrite, but also there are environmental issues of eutrophication, nitrous oxide gas emission, and you name it. So nitrogen is always the cycle that is mostly dealt with aquaculture, and that's the equilibrium here. So everything starts as NH3 in aquaculture system because when the protein breaks down, it breaks down into ammonia. So always it gets into the water, and it's an equilibrium based on pH, okay? So at normal pHs, 7 or 8, mostly is in this form of ion. So what happens is that NH3 reacts with water and turns into NH4 plus OH ions, and alkalinity is increasing, okay? So ion is a base. Ammonia is a base, yeah? It can be used as a buffer often. So that's how it all starts. And obviously, the higher the pH, the more ammonia we have, which is the most the toxic form. And you can see that on many fishes, it depends whose, but ammonia as an ion, yeah, 1 milligram per liter or less can already be toxic and affect the nervous system, okay? Can cause hyperactivity and lethargy and coma and mortality, and just you name it, it's there. But there are systems, at least we're working on such systems in Israel, not so much me at the moment, but that we can grow, if you control the pH and make it 6, for example, in these ponds, then you can make sure that 99.9% of your ammonia is in this ion form, which is much less toxic, and then you can grow fish at 20 or 30 milligrams per liter of ammonia in the system, which is very non-typical to fish growth, which is usually up to 3 or 4 or 5 milligrams per liter. Okay, so that's the with the toxicity, and part of the reasons that the reason that ammonia is toxic, because it's a decoupler. It's in the ATP process. It prevents formation of ATP. This is part of the deal, and the toxicity comes from the fact that it's unionized and it can go through the gills and through the membrane, cell membranes easily. Flow without any barrier. There is no barrier there in the gill or in most cells. So when the water concentration of ammonia is high, you get ammonia getting in and doing all its bad things in the blood and changing the osmotic pressure. There is an active mechanism in the fish that can take ammonia out, and replacing it with sodium, but that's limited, and that's where it's becoming toxic, less or more, and that's where the fish can tolerate a certain concentration, but from a certain concentration, it's just affected. So most systems of aquaculture use nitrification to remove ammonia toxicity. And as again, that's something for you all to think about, is whether that's the best way of doing things or not. There are other means to remove ammonia, but that's something that I think aquaculture will have to develop, because we as a group of aquaculturists are quite captured by this process. So you know, I'll show that, but basically NH3, right, plus, God, anyone can see something, NH3 plus oxygen, right, gets to NO2 and then to NO3, and it's all bacteria mediated, different groups of bacteria, and that's also not true anymore. There was, I think a month ago, there is a paper in Nature, look for it, it's interesting, where one group of bacteria can do the whole thing. So it's not splitted anymore. I don't know how important it is, but there are definitely bacteria that can convert ammonia to nitrate as one, just one species and not the two groups. So these are called AOB, right? Ammonia oxidizing bacteria, and these are called NOB, nitrite oxidizing bacteria, right? And recently, in the last few years, we also know that some archaea can do that, and not only bacteria. But again, their role and their importance is not really known. So let's stay with our current knowledge and say that these are the two important groups and the important ones. So this is an intermediate, and since we are talking about two groups, this group has its own requirement, this group has its own requirement, and really it's not always balanced. When it's not always balanced, we have accumulation of NO2. NO2 is also toxic, and why it's toxic? Because it competes or it converts, it's absorbed on the gills and decrease the affinity of oxygen to the hemoglobin, okay? And interfere with the oxidation reduction of iron in the hemoglobin. And what it causes the hemoglobin to be converted to a metahemoglobin, okay? Because the iron is oxidized. And then the blood turns brownish, and that's why it's called brown blood disease. You're definitely familiar with it if you grow fish. And low affinity to oxygen, respiratory stress, and they die, the fish. And chloride, being an ion of pretty much the same size, diameter of nitrite, NO2, is competing on the metahemoglobin sites. So if you put enough chlorides at a 1:10 ratio, 10 times more chlorides, then the toxicity of NO2 is being reduced, but still anywhere between 0.3 to 2 milligrams per liter are considered to be safe limits for NO2 concentration in the water, and not much more than that. To break down the list, there is list of parameters, just an experiment that was conducted a few years ago about the toxicity of nitrite, and again the importance or how do you determine toxicity, what is toxic, how you do that, which is a bit tricky in these systems, but what try to demonstrate here that when you have an input of feed, you basically creates a load of ammonia into the system about an hour or a couple of hours after it is introduced. So if you have a good nitrification filter, you know, the ammonia will rise and immediately will drop and convert it to nitrate. If you don't have established filter, then, you know, you'd have these different peaks. It would not work very well. And that's something to remember when you start. The same thing with nitrite, because of the whole system is dependent on these bacteria. So when you have an established biofilter, nitrite is not accumulating, but when you do not have an established filter, nitrite is accumulating. And when we're looking at the toxicity effect, what we saw here is we grow fish at different concentration of nitrite, you know, 0, 1, 2, 4, and 8 milligrams per liter. And we grew shrimp in this case. That's why survival is quite low. And we looked at percent mortality. So, you know, when we looked at, when we grow the shrimp at 8 milligram per liter, you know, after very few, after a couple of days, they all mostly died, you know. So that was definitely toxic. When we grew them at 1, 2, and 4, you know, they were pretty much behaved like like no nitrite at all in terms of mortality. But when you're looking only at mortality, you lose the picture because it's how it affects or how it's, you know, the performance of these shrimp. And when you look at the performance of the shrimp, you see that already at 4 milligram per liter, they just don't grow. So they don't die, but they don't grow. So since it's aquaculture, we want them to grow. And at 8, obviously, the net production is minus because mostly they died. Really, the important thing is when you determine this toxicity level is how you do it, and you'd have to look at the performance and not only at the LC50, which is the traditional way to determine toxicity. Okay. LC50, lethal concentration of 50% of the population. Okay. And then we turn everything to nitrate, which is less toxic or the least toxic, and concentration can get up to 1,000 milligram per liter. Yeah, that's sometimes the lethal concentration exceeds 1,000 milligram per liter. In recirculation, we're trying to keep these systems under 100 milligram per liter of NO3 as N, okay, not NO3. Remind you, if you look at the weight of NO3, it's about 5 times higher than just the N, yeah, the ratio. This is 14 and this is O3, 16 times 3, 48. So it's, the ratio is 14 to 60. So it's 4.5 if I'm not mistaken. And just to complete this shop list, I just gave you from various books of aquaculture books the recommended concentrations of different parameters in the pond, and obviously it changes from pond to pond, from fish to ponds, RAS. But I mean, as a general rule of thumb, I think it's a good thing to have and keep in mind. Okay. Surviving still. So yeah, I guess we'll continue. Otherwise, we'll not get anywhere. So drink water. So what we said is that when we look at this aquaculture, so now we basically set up the standards or the goals, the targets that we want to get with water quality, okay? Now we need to get there. So initially we have the initial water that we get, but we introduce this feed and we have to make sure that we meet these standards. And that's why if we don't want to use water raceways to wash the water, then we have to treat it if we're recirculating it, and that's all. This workshop is all about recirculating aquaculture system or aquaponic system, which is recirculating. And we want to reduce this water exchange. So we have to treat the water. And obviously, it's a trade-off between exchanging and treating, and then it's an economical issue. Okay, so we said feed and water are the are the only inputs. But water, since it's recirculated, it's a small input, where feed is the larger input in terms of every parameter that we're measuring. And excrete ammonia, the fish, which is what we will look at, and excrete carbon. And then we're talking about removal of solids, and that will be the next part of the day. But we remove the solid from the fish area, and then we'll treat it, and then we nitrify it. That's 99% of the systems. Okay, and as we said, nitrification produces acidity. So for each mole of ammonia, we produce 2 moles of acidity. That's important. Remember, though, that it can be somewhat balanced, because we started with NH3. So when it started with NH3, we produced OH. So 1 mole of hydrogen already is being consumed by the alkalinity that we produced. So we're just in a 1 hydrogen excess at the system. Now, when we're looking at these systems, as we said, we wanted to look at it as different treatment loops. So we have this unit, and we have this bit, and we have this bit, for example. Okay, it's all different operations that can work at different rates. And for that, what we need is, if we want to quantify these things, we need to look at water exchange and balances, and get into a little bit the very basic of engineering. So I don't know what's your background, but I decided to put few of these slides just to go again to have a baseline. So when we're talking about water exchange rate, yeah, it's the water flow into the system in terms of volume per unit time, and it obviously important, the system volume. The larger the system, the longer the retention time for a certain Q or flux. Okay, so exchange rate is the Q over, or the flux over time, divided by the volume. And if we want to talk about exchange rate in percent, then it's just the time 100. So if we had a flow rate of 300 cubic meter per day and a pond volume of 600 cubes, then the exchange rate would be half a day, yeah, or 50% exchange rate, and the unit would be 1 over day, or 1 over time, yeah. If we have the other term, which is related, is the retention time. So we can talk about how much water we exchange, or how much time water stays in this system. So if we had, you see, 2 reactors, 2 ponds, and this is the Q, the inlet is 10 cubic meter per day, and go into another reactor and then go out, then the retention time would be V over Q. Pay attention, just the opposite, the reciprocal of the exchange rate is the retention time. Yeah? So it'd be 10 days for this tank. It'd be 5 days for this tank, yeah, because 50 divided by 10, and it would be, the overall retention time would be 15 days, okay? So, and if we want, the retention time. So if we want to talk about exchange rate, it would be 0.1 per the first volume, 0.2 for this tank, and the overall it'd be 0.067 because it's the total volume. So that will be the exchange rate, okay, over time. If you have questions, definitely ask, because that's an important thing. Maybe I want to just, no, I'll do it later. That'll be the next. Okay. So then we can start talk about the recirculation, yeah, the recycling ratio, how much water recycled through these different tanks. So that would be the Q, again, the flow, the flux into, divided per reactor, yeah, divided by the overall, or by the total flux, yeah. So, or this other container that you were interested. So if we have a Q of an inlet, yeah, flux into a certain volume, into a certain pond, and some water goes out, but we now recirculating water through these treatment systems of ours, yeah, as a separate loop, which is not necessarily working at the same rates as the main tank, okay? So if we're looking at the retention time of here at this pond, it be V over both Q's, okay? Yeah, and it would be 0.9 days, the retention time, yeah? And the retention time here would be just the volume over the Q that gets into this system, okay? So it'd be only half a day, okay? And the retention time, the overall retention time would be 15 days as we were before. You remember, because it's 150 cubic meter when you introduce 10 cubic meter a day. So overall, it'd be 15 days before we exchange the water. That's simple. And we can also talk about the retention time only of this tank, but that's not, just it's there. So it's the same thing with the exchange rate, but now it's the other way around. It's reciprocal, yeah? It's Q over V, not V over Q. Now it's going to be the exact same thing, okay? Now it's 1.1 for the exchange rate, I mean for the exchange rate here, for the exchange rate here, which is different, only 2 and, 2 per day. The exchange rate here would be 0.067 per day and so on. So that's an important thing when you want to start and look at balance. How much gets in terms of nutrients, how much gets out, and what's produced and what's recovered. So for each one of these tanks, there are inputs, there are outputs from the tank, and there are things that is either produced in the tank or removed from the tank, okay? So this is what happens here. We have input of water with food or nutrients or whatever, inputs of something, of nitrogen, which is dependent on the flux in, yeah, cubic per time, and the concentration that we start with. There is always a flux out, and possibly a different concentration that goes out. There is removal of things because there are chemical processes occurring, and there is production of things. Ammonia is produced, CO2 is, produced, and other things are removed. I don't know, when you have denitrification, nitrogen is removed, okay? And if you want to put all these mass balance equation in terms of a simple equation, then you'd say that whatever accumulates equals to what we introduce in plus what is produced minus whatever goes out, which is output rates in the flow or removal rates in these chemical processes. So, so if you want now to put it in an equation, again, the simple version of this equation, it will be volume, yeah, the difference in the change in concentration over time equals, so that's the accumulation rate, yeah, this is in words, this is just in math. Flux times concentration in, whatever produced, flux out times concentration, whatever removed. So it's exactly the same thing, okay? And if you want to look at the units, then if we're looking at concentration of something, the flux would be cubic meter per hours, concentration would be milligram per liter or gram per cubic meter. Production is gram per hour. Removal gram per hour, flux time concentration. Anyways, the balance, the accumulation rate is unit of mass per time. Now, if we're in a steady state operation, that means that there's no changes. Ammonia is fairly stable. Yeah, there's no changes in concentration. Then basically no accumulation occurs. So this term is really zero. There's no accumulation. Okay, so it's zero now equals the flux in concentration with production, blah, blah. It's the same thing. Okay? Now it simplifies our life. And then if we rearrange this equation, then it turns into, if this is zero, so you see we can take the Q out from, for the parentheses, and we can, yeah? So if we're looking at oxygen now, okay? So this is zero, Q is here, DO in minus DO out, okay? Minus whatever is removed. Okay, so because there is no production of oxygen in this case, so production is also zero. Okay, so that's an easy equation to solve, but then it turns into be like Q is equals the removal of dissolved oxygen minus the difference is whatever gets in and that gets out. And if we take an example, so even if you, it's hard to follow it the first time, you have it with you. And it's a very, very simple example. It's like if we start with a DO of 7 milligram per liter with the input water, yeah, there is a flow-through system. Yeah, the water here is 7 milligram per liter. The water out is 5 milligram per liter because the fish is consuming oxygen. Then we ask ourselves what should be, or we want, if our design target is that the concentration in the outlet would be 5. Yeah, we start with 7, and we don't want it to get below 5, because that's what we decided. If we wanted, we can decide that it's going to be 3. So we say, what should be the flux of water in order to maintain this oxygen? So we can start getting numbers, quantity. That's the whole, what I'm trying to get here, that it's not like that. We'll build up a 1 cubic meter tank, put some fish, we need settler. Let's just put a settler, beads, calculate it. Okay. So in yeah, in this example, the fish consume 200 grams per hour. Okay. So we know that the Q, the flux into, has to be 100 cubic meter per hour in order to meet our constraints of 500 milligram per liter in the outlet, yeah? Because the Q, which is our unknown, equal, and we just saw it earlier, equal to this removal rate and the difference in DO concentration, dissolved oxygen concentration. And it goes on with a few such examples. In this case, we want to figure out what would be the removal rate. So that's the question. What is the removal rate? So instead of looking at the Q that we asked here, we now want to look at the removal rate. Okay, so what have I done wrong? Here. So we want to go look at the removal rate. So we rearrange this equation, and we know that the removal rate is equal to the flux in minus the differences in concentration. So if we know that we introduce 6 milligram per liter, that's our given, and our concentration at the outlet is 5 milligram per liter, and the flow rate we know because we introduced, what is the removal rate? We introduce it into this equation, and we see that it's 15 grams per hour. Okay? So this is the idea, and it can get also with chemical reaction, okay? Like, for example, ammonia, and with that, I promise that I'm going to stop. So ammonia concentration, for example, is so and so in the pond. With our influent water, we have none. The influent water are flowing at, for example, this thing at 70 cubic meter per hour. So what would be the production of ammonia? That's our question in this case. It'd be, 98 gram per hour. We can now quantify that. We know how much we need to treat. We know the quantity. Okay, so we know how many bacteria we need there, how much biomass, what's the sizes, what's the aeration that it's going to take us to get there. And that's the idea. So let's now look at the nitrogen cycle a bit more closely, and that's what we're going to do in these labs. So I just picked up, something from the internet. It's very, very schematic nitrogen cycle. Nitrogen cycle is a very interesting one because it really has chemical reactions, biological reactions. It's solid, dissolved in gas. It's a brilliant cycle, and quite a few people are studying it for many years, built various careers on that. So in this case, we have fish, we said, where they excrete ammonia. This is our source. Mostly we said nitrification to nitrate, on the way if we want to remove nitrogen, we denitrify it mostly. So to denitrogen gas by either denitrification or anamox or different processes. But on the way, we have some nitrous oxide emissions to the atmosphere, which is another challenge, environmental challenge. Here in the process, we have some nitrous oxide emission, which is another challenge. This nitrogen, if we're talking about aquaponics, can go to plants, or ammonia can go to plants. Is it important or how important is to set a ratio? Ammonia can have this non-infectious disease in tomatoes, in peppers. These, I don't know the name of these deformities in English, but there are deformities if you irrigate only with ammonia. So there is a certain ratio per plant. So how do we control these things? Can we control these things? And of course we can. It's just a matter of, again, the amount of effort and the amount of productivity that we're going to gain or lose. So these are the kind of questions that we're asking when we're trying to look at these issues and at the treatment processes. So nitrification, we talked about the process. You're all familiar with it. So again, let's assume that we have these two groups of bacteria, ammonia oxidizers turning into NO2. Again, pay attention that we produce hydrogen. And by the way, I don't know, maybe it's very, very trivial, but I thought it's a good point to say that the bacteria is not working for free. They're working to get energy. Yeah, that's what they are doing. They're not doing it because we pay them or, we talk nicely with them or we hit them. No, that's not the way it works. They're working for energy, to proliferate, right? So it's all about oxidation and reduction. It's all about electron transfer. Electricity is electron transfer. Yeah, if you put a battery, you move electrons and you get light. The same thing with bacteria. Yeah, so pay attention that it's all about electrons and about energy. I know it's trivial, but I know that we often tend to forget that. And then there is this process, the second process, which is nitrite to nitrate. And you can see that it gains somewhat less energy. So bacteria would, this process is likely to occur faster, or the driving force is greater than this process. That can create this imbalance nitrification at times, something that you can, and then accumulation of nitrate, which is not good for our system. So that's something you need to remember, and it's an interesting point of view. So it's not the bacteria or that, it's energy. It's all about energy, yeah? All this word is moved from energy. No, it's all about oil and things like that. And denitrification, removal of nitrogen, is basically taking this nitrate and convert it into N2 gas, which is inert. So these bacteria needs carbon dioxide. They're autotroph. They're slow grower. Okay, that's very important also to remember, because they're often outcompeted by these other bacteria, specifically in these systems that has a lot of organic matter in it. So we have to make sure that we maintain the right conditions for these as well. So it's oxygen and inorganic carbon, where denitrification is anoxic. Anoxic means that there is no oxygen, but this nitrate is the electron acceptor, okay, instead of oxygen. Where anaerobic means that there is no nitrate and nitrite, okay, so that will be considered anaerobic. And everyone is mixing these terms, but doesn't really matter, just so you remember. So they need organic carbon, ammonia, and nitrate to convert it to biomass, to bacteria, and they produce CO2, and they produce N2. Okay, and if we look at just a typical equation from aquaculture, you know, then organic matter, this is citrate plus nitrate, yeah, is converted into biomass, water, CO2, and nitrogen gas. So that's what happens there. Now, the way these bacteria are working, the rate that they are working, follows one of several rate processes. Usually it's either a zero order or a first order. Let's leave this second order thing. What does it mean? It means that as the concentration of the substrate, if it's denitrification, it's nitrate. If it's nitrification, it's ammonia, is either affecting the rate. But if we have a lot, if we have 100 milligram per liter of ammonia, I don't know, 50 milligram per liter of ammonia, or 30 milligram per liter of ammonia, the bacteria would not care. The difference in concentration would not change much, would not change the rate that they are working. Where if we had half a milligram per liter of ammonia or 10 milligram per liter of ammonia, then there would be a change, okay? So then they'll work at a first order kinetic. So basically that means that there is no effect of the concentration, where here there is an effect of a concentration on the rate that the bacteria run their process. And if you combine this, first order and zero order into one system, you get the monod relationship, or the, what's the other phase? Michaelis-Menten, an enzymatic relationship. Yeah? So at the lower concentration of your substrate, it's a first order kinetics, concentration affecting the rate. At the higher concentration, it's not affecting the rate. Now there is a place here where we call it the Ks, where we determine that above this concentration, we're moving into the zero order, and below this concentration, we're moving into the first rate order. And that's very important when we design our systems. Okay? So this Ks is really important, and the maximal rate of process is also very important. So these two parameters we want to find. And we did that. I mean, that's what we're not going to do it. Well, we're in the lab today, that's what we're going to do. But since we don't have enough time and we're not going to get reading over time, but only 2 readings, we will not be able to establish the figure. But that's the way to establish this figure and get these rates. Okay, so that's what we want to do. So as we said, we start in a first order, and then it goes into a zero order. There would be a Q. Q would be the rate of change. Nitrification, how much nitrate is produced or how much ammonia is removed over time, okay, per volume. And we'll find this value or the concentration of ammonia that below that it will affect the rate, and above that it will not almost affect the rate, this Ks, which is half of the maximum rate occurring. Okay, that would be the Ks. So the maximum and the Ks. Okay, and if you want to put this, if you take a solution at different concentration, I don't know, 1, 2, 3, 4, 5 milligram per liter of ammonia. Yeah, and we will look at the rate is being transferred or converted to nitrate. We will get different rates at the different concentration. We can establish this graph and then find out what are the two parameters, the important parameters. So that's what we're going to do today with nitrification and denitrification. Okay. Now, if you if you find the equation of this graph that is empirically conducted, it will follow this: the rate, the nitrification rate, denitrification rate, is equal to the maximal rate times the substrate concentration, ammonia, nitrate, whatever, divided by the substrate concentration and the plus the this Ks. That's the graph equation. You know, when you plot it on the Excel today, you don't have to work hard. And what we can learn from that is the following: that if the substrate concentration, that's where I was aiming, if the substrate concentration is very low, it's lower than this Ks here, then look, the substrate concentration can be is negligible, you see, and then we get that the rate would be equal to Qmax and Ks, which are constant. We found them; they are constant, times the substrate concentration. So we can, you know, we can write it like that. At low concentration, we have some sort of a factor times the concentration of our factor, and we can determine the rate of nitrification in our system, or denitrification. Where if the substrate concentration is higher, and that's more intuitively, then it's here, then we get the maximum rate. Yeah? Why? Because if the substrate concentration is high, then Ks can be eliminated, and then we have these are also eliminated, and we have that the rate equals to the Qmax. You can see it here. Okay, and that's important again to remember. So the other mechanisms of nitrogen cycle, so we have nitrification, we had denitrification, and basically in aquaponics we have assimilation. That's the third mechanism that we're using. In this case in plants, but we're not gonna, oops, we're done almost. We're not gonna take care of it so much here, but again, I mentioned the ammonia nitrate concentration. Okay, what the ratio, what type of ratio? So the preferred ratio, that's for tomatoes, is about 1. It's changing with plants. And you can also know how much, you know, would take you to grow, what would 1 kilo of nitrogen will support. So now we can start, plan our systems, okay? And now, basically, we can either take a few minutes break. I want to start the lab here to organize it, basically, to talk about the methods, not in the lab because it's busy, and then we can move to the lab. Maybe we can take a 5 minutes break or 10 minutes break and then talk about the lab.